Hello and welcome to Research Matters Podcasts. Today I, Aishwarya Viswamitra, welcome you to part 2 of our series on the research behind the Nobel Prizes of the year 2020. In this episode, we discuss the Nobel Prize in Physiology or Medicine. The 2019 winners, William G. Kaelin Jr., Sir Peter J. Ratcliffe, and Greg L. Semenza, won the Nobel Prize for their discovery of how cells sense and adapt to oxygen availability. This year, Harvey J. Alter, Michael Houghton, and Charles M. Rice were awarded jointly for discoveries that led to the identification of the hepatitis C virus. The word hepatitis comes from the Greek words for liver and inflammation. Hepatitis can be caused by a variety of factors, including autoimmune diseases and alcohol abuse, but the most common cause is viral infections. This is known as infectious hepatitis, and there are two kinds, hepatitis A and hepatitis C. While hepatitis A spreads through polluted water and food, it is preventable by a vaccination. The symptoms are mild and the long-term impact is little. But the other infectious hepatitis, hepatitis C, isn't as placid. It is a blood-borne infection, which means two things. One, that it is transmitted through blood, and two, that a seemingly healthy individual could be actually silently infected for many years before serious complications arise. Unlike hepatitis A, this form of hepatitis has serious long-term effects and complications like liver cancer and cirrhosis, which is also a form of liver damage. Considering that this form of hepatitis has caused more than 1 million deaths per year worldwide, you can imagine that developing vaccinations and cures have been the top priority in hepatitis research. However, this form of hepatitis is caused by not one, but two viruses, hepatitis B and hepatitis C. The first step to developing a vaccination or a cure is to know what you are up against. Thus, identifying the causative agent and its biological and chemical structure is of primary importance. In the 1960s, Baruch Blumberg identified the hepatitis B virus. This discovery led to the development of diagnostic tests and an effective vaccine, and he was awarded the Nobel Prize in Physiology or Medicine in 1976. At that time, the story of blood-borne hepatitis infection was thought to be over. But soon it became clear that the hepatitis B vaccine only accounted for a portion of the blood-borne cases. There was another novel virus at work. Nobel laureate Harvey J. Alter was behind this realization, and along with his colleagues, he showed that blood from these hepatitis patients could transmit the disease to chimpanzees, the only susceptible host beside humans. This was an important observation because now the disease could be studied in a non-human host. Meanwhile, Nobel laureate Michael Houghton was working at a pharmaceutical firm when he began his quest to discover the elusive virus. Along with his co-workers, he created a collection of DNA fragments from nucleic acids found in the blood of an infected chimpanzee. Though most of the DNA fragments would be from the chimpanzee's genetic code, Houghton hypothesized that some would be derived from the unknown virus. But how could they be certain which the viral fragments were? Well, they knew that antibodies are produced in response to a viral infection in an infected individual. So, Houghton and his colleagues obtained antibodies from infected patients, and they used this to match and identify the viral DNA fragments. Further work showed that these viral fragments were derived from a novel or new RNA virus, and it was named hepatitis C virus. But one question remained. Could the virus alone cause hepatitis? To answer this question, the process had to be reversed. Rather than studying deceased individuals, the scientists now had to determine if the virus could replicate and cause the disease. This work was done by the third Nobel laureate, Charles M. Bryce. This final breakthrough proved that the virus can cause the disease on its own and was published in 1997. Much has happened since then. With knowledge about the virus's structure, highly sensitive blood tests for the virus have been developed. Previously, blood donation would cause a major percentage of the hepatitis C cases because of blood transfusion. But now with these tests conducted beforehand, post-transfusion hepatitis has been essentially eliminated. Not only that, antiviral drugs have been developed, and what's more, a cure has been too. Eradicating hepatitis C from Earth's population is now possible, and efforts to do so have already begun. Thank you for listening to this episode of Research Matters Nobel Prize series. Stay tuned for the next episode coming soon. Till then, for more such science news and podcasts, please log on to www.researchmatters.in.